Okay, so we're nearly done. 5 minutes. So then this is like a plug to one of the projects that we're working on. This is ARCHS4, where we try to run RNA-seq on all publicly available data. So so we do this for human and mouse, and then some other species that we have in something called ARCHS2. But there is like trillions of reads that we aligned in the past. So this is way more than a petabyte of data. So 1 million gigabytes. And it's probably more like 3 million gigabytes or something. It's like a huge amount. And yeah, so there's more than 1 million samples out of 300, like 30,000 different experiments. So it's a really like comprehensive website. And here you kind of see how the data increases over time. And so this project really focuses on minimizing, CPU cost, memory, storage, and network bandwidth. So it's a more like a networking problem than anything else. And so how it's designed is that it kind of runs in the clouds. We use AWS, which is like the Amazon cloud. And we have all our jobs kind of Dockerized. So they run these, we're running Callisto in this like Dockerized container, and it basically fetches instructions from a server that we have. And that instruction will basically contain information about, it's just a JSON file from an API, and it will tell it where to get the file, how to download it, and then where to copy it once it's completed. And so when we run these in batches, we run, easily like 400 or 500 of these instances in parallel to process all that data that's out there. And yeah, as I said, so this is already out of date, but so we have, nearly, like it's way more now. I think we have we're getting eventually close to 1 million for human and 1 million for mouse. So we're still a little bit under that, but pretty substantial. And then about 35,000 from other organisms, but much less. And then so the nice thing, once you have like such big data sets, you can really get a really good idea of the landscape of gene expression. So you can just look at different cell types or cell lines, and then you can really drill down and say, for this gene, what is it expressed in which tissue and what cell line and whatnot. And so this going all the way back to the initial plot really can inform you about, the mechanisms that underlie biology. And we can kind of ask, given different cellular contexts and gene expression of a gene, what processes it belongs to, so we can annotate genes just based on the gene expression because it's an unbiased data set. So this is the good thing about RNA-seq. It's really unbiased, right? It's a very objective measure of what's going on in biology. So it's ideal if you want to annotate it with like something literature-based, because it's a data-driven resource. So it complements each other really well. So we have these gene pages where you can look at predictions of what genes do just based on the gene expression. And then we also had like a new version, which is called PrismX, which is slightly smarter than this, but it's kind of the same idea of predicting, functions of a gene. So there's a lot of online resources you can look at. I just put in like the ones that I think are like the most interesting if you want to check them out. And there's a bunch of citations. If you have a lot of time, you can go through them, but this should cover most of the things that I mentioned today. So yeah, just on time. So yeah, I want to acknowledge the lab. So Avi is the PI, and then a lot of the people here on the list, I think, will give lectures in the next couple of weeks.